Hi everyone, my name's Shelly and I'm a grad student here at Salk in the plant biology department. There's about 70 people in our department, and as you can imagine there's a lot of different projects going on. So you may be wondering, why do we study plants and what do they have to do with us? Well, one major reason is because plants provide the majority of food that we eat. All the produce and grains that we eat and the animals that we eat are also fed from these plants. And there's also a growing world population, so we need to keep growing a lot of plants to keep the world fed. And climate change is also an issue where plants are having a harder time growing in their normal conditions because things are changing. So here we're really trying to understand how plants grow best so that we can most efficiently grow crops and be able to feed the growing world through a changing climate. So besides all that, plants are just amazing and super resilient organisms because unlike us, they can't just get up and get a glass of water when they're thirsty or go inside when bad weather comes. They have to deal with all these changing conditions just using the cells that they have. So a big question that all of us plant biologists are trying to address are how are cells responding and what specific cells are responding to make plants so resilient. So why don't we go take a look at some of the things that we do here at Salk to understand how these plant cells respond. So this is one of our greenhouses here at Salk. You can see it's restricted in the amount of space that we have. It is still pretty big, but we can't be growing large fields of crops in here. So instead of growing crops, we grow this plant called Arabidopsis thaliana. This is what it looks like. It's actually a weed, and the reason we do plant biology on this plant is because it grows a lot faster than crops do. It has a much simpler genome, and it's just a lot easier to work with in that it doesn't take up a lot of space in the lab. So this is the wild type Arabidopsis that is used as a reference that a lot of probably the most amount of biology is known about this particular plant. These are some other types of Arabidopsis that we study. This one is an English variety that comes from Great Britain, and this is another type that comes from an island that is very tolerant to hot conditions, but not as much to cold conditions. And this is yet another type that has a mutation. So this is the Arabidopsis columbia. It has a mutation in the MET1 locus, which is a gene that controls gene silencing in the whole plant. So you can see the leaves on this are very different than the leaves on these plants. And by studying all these different types of Arabidopsis, we're able to ask questions about how the differences in these plants' DNA affects how they grow. And one of the really cool things about the Salk Institute is that we house this big seed collection, which is essentially a library with a lot of different Arabidopsis varieties that have been collected from around the world, and also a collection of mutants that are plants that have mutations at every single gene in the genome. So there's about 30,000 genes in Arabidopsis, and we have this collection where there's all these different mutations in these genes. So we can ask questions like this plant, what about this mutation is making it have this effect? So let's go check out what the seed room looks like. So this is our seed room here at Salk, and all of these boxes and envelopes contain different types of Arabidopsis seeds. This is one of the biggest collections in the whole world, and researchers from all over will contact us and the seed banks that we send these seeds to get these seeds to do experiments with. Each box contains all these different vials that have different barcodes on them, and each one is a different plant. These ones are different mutations. Like if we want to study what are the effects of a mutation on a certain gene, we look through a catalog that we have, find the tubes, and then grow the seeds up and do our experiments. So one thing about doing plant experiments, in the greenhouse that you saw earlier, that was an open air greenhouse that allows different weather to come in and temperature changes to happen. So like This room is very temperature controlled, humidity controlled, and that's to preserve the seeds. In our experiments, we also want to control those environmental variables so that when we grow plants, our mutant plants, or our different ecotypes, we know they're growing, like the conditions that they're growing in, so that the effects we see in the appearance of the plant is not due to these changing environmental variables. So let's go check out a controlled growth room that we use to do our plant experiments in. This is one of our growth rooms here at Salk, where you can see there's no windows, and all the light that's coming in here is provided by these fluorescent bulbs at all these different shelves. And these bulbs are on timers to simulate a 16-hour day and an 8-hour period of darkness. That way we can really control what these plants are experiencing. And these are some of the experiments that people are working on here. You can see these ones are much smaller because they're younger plants. And one specific thing that we're studying here at Salk is how do plants respond to shade? And the reason is because when we're trying to grow crops, we want to grow them really densely so that we can grow as much as we can in the space that we have, but they end up shading each other, and that causes loss in productivity. So one thing that we're studying here at Salk is how these plants respond to shade. And that's because when we grow crops in the field, we're trying to plant them at a really high density so that we can grow as much as we can in the space that we have. But when you grow plants really close together, they start to shade each other, and that can cause a loss in productivity. So to do these shade experiments on Arabidopsis that we're studying, we actually use these light chambers where we can really fine-tune the wavelengths of light that the plants are getting exposed to. So let's go check out what those light chambers look like. These are the LED light chambers that we use to do our very finely controlled light experiments. We can mimic shade by changing these different wavelengths of light. So there's these different controls here, so temperature and the different wavelengths that we can change. And inside, you can see in here, there is red light and blue light coming through. So we can actually adjust the settings on these to exactly mimic the light that plants experience when they're in a shaded condition. You're probably wondering why this room is so green. This is our green room, and we use this to do experiments on plants when we don't want them to perceive any light. So plants can't actually see green light. They're completely blind to green light. And that's why they actually appear green. They reflect it. They don't absorb it. So when they're in here, they're experiencing complete darkness. And this helps us. We do experiments on plants in these conditions. Like if we want to spray them with pathogen or treat them with different plant hormones, we can do that in a dark setting. So when we see how the plant are responding, we know that it's not coming from pathways that are stimulated by light. Another technology that we use here to study how genetic mutations and different treatment conditions affect plants is by using confocal microscopy, and that's what Carl here is doing. He's imaging an Arabidopsis root tip that this particular plant has a green fluorescent protein reporter that senses auxin levels in the cells. And what you're seeing is that there is a lot of auxin that localizes in the root tip here. And that's because auxin is this super important plant hormone that's involved in all stages of plant development. And so what we can use this technology to do is find out how this plant process localizing auxin to the root tip works in a normal plant, and then study how that's different in a mutant plant or a plant that's treated in a different condition. To go beyond the cellular level, we use other techniques to study actually the DNA in the plant cells, and also their transcripts, and and methylation patterns on the DNA. And to do that, we use next generation sequencing. And I'm sure you've heard about sequencing your genome in 23andMe, where you can get your entire DNA sequence. So we also use those kind of technologies to study what's happening in the DNA level and the gene expression level in the plant. So I'm going to go show you what tools we use to do that down in the other lab. These are the Illumina sequencers that we use to do all of our DNA, high throughput DNA sequencing here. And they work by fixing. What we do is chemically fix DNA onto these flow cells, and then these machines have a super high resolution camera that's able to photograph different fluorescently labeled nucleotides. So you can see here there's different images that the camera has taken. These are all the fluorescently labeled T's, G's, C's, and A's. And so these photographs happen sequentially, and at the end of the sequencing run, the computer compiles all of those images and outputs all these sequences of DNA that we then analyze. But how do we get DNA from a plant onto the flow cell? Let's go take a look at how we do that. Actually, just going right here. So first what we would do is take our plant, maybe like the ones that you saw in the growth room, and put them in liquid nitrogen, which we store in these containers, and that freezes them so that we can grind them up in the mortar and pestle, and then we use these kits and a series of different buffers bind the nucleic acid to a column like this to purify it from all the other plant material, and then we end up with material that we then have to do this thing called a library prep to, and that's just a series of different reactions that get the DNA ready to put on the flow cell. And sometimes we have a lot of different samples that we need to do at once, so that we only have 2 hands, and it's really would take a long time to do that many samples. So sometimes we use these liquid handling robots to help us prepare a lot of samples at once. So let's go take a look at how those work. This is the liquid handling robot that we use to help us prepare a lot of library preps at once. So instead of that one tube that you saw me holding before, these plates have 96 different wells in them, and it's like doing 96 samples at once, except that I don't really have to touch anything except the mouse button on the computer. So it helps save us a lot of time and can help us get our samples ready for the sequencer a lot faster. Once these samples are prepared and ready to go on the sequencer, it takes us about a week, sometimes less, to get the data back. And when we get the data back, we have to start doing all different kinds of analysis when we look at it. Let's go see what some of that analysis looks like. When we get our data back from the sequencers, we upload it on the computer, and we can look at it in a genome browser like this. This genome browser is showing data that came from the wild-type plant Arabidopsis columbia and CBI, which is the island plant that you saw down in the greenhouses. And here we're looking at one gene where in the wild-type plant it's not expressed, but in the island variety it is. Maybe this gene is an example of how this plant is able to tolerate high heat conditions and not so much cold conditions, and how the columbia is different. Well, I hope you learned about a lot of different ways you can do plant science and how we can understand plants so that we can keep the world fed. Thanks for stopping by.